Today I'm going to talk about native Hawaiian plants to increase Hawaii's agricultural production. So my talk will be on landscape and indoor plant studies that we've been doing for research. So why native Hawaiian plants? Promotion and use of native Hawaiian plants have been increasing in the last 20 years, and this has been partly due to issues for biosecurity, particularly invasive species and conservation issues. And also we have legislation that requires native plants in state-funded landscaping projects. Aside from state-funded landscaping projects, native plants are also now being incorporated in roadside rights-of-way areas, particularly here in Honolulu. So in the next 13 years, the demand for native Hawaiian plants is expected to increase. This is because of the passing of Act 233 relating to Hawaiian plants. And this is a state law that increases native plant footprint from 10% by 2019 to about 35% by 2030. So despite the increased acceptance and use of native Hawaiian plants in urban landscaping, there's still very little progress in terms of developing selections in nativars. So here at UH Manoa, we started an native ornamental plant research program in 2015. And our program aims to select, collect, and evaluate non-endangered and underutilized native Hawaiian plants for the following purposes. So we are currently looking at native Hawaiian plants, trying to expand the variety of native Hawaiian plants for landscaping. And then we're also looking at uses for potted flowering and container use, as well as for indoor foliage and cut foliage. So the current species that we're looking at for native plants for landscape use are the following. So we're looking at evaluating Pacific lovegrass or Aerogrostis deflexa, Alima or Cydaphalax, and Aveo, Chenopodium oahuense. So Chenopodium oahuense or Aveo is a drought-tolerant shrub that's found on all islands. We have selected a prostrate and compact forms. And these came from the island of Molokai. And we're kind of looking at it as a potential ground cover container plant or a green wall plant. And typically, Aveo can be propagated from seeds. However, you can get some seedling variation. So we're kind of looking at cuttings as a way to propagate it to maintain the growth form. So the research that we're doing is propagation and landscape trials. And this is just what we have done in the last year in terms of looking at propagation studies for this particular species. So we tested the application of rooting hormone, see if it improves rooting of the two selections and if there's any differences in rooting behavior between selections. So we tested compact and prostrate selections with or without rooting hormone. So no rooting hormone, 0%, 0 ppm IDA, and Hormex-3 is our hormone treatment. And we put this in a mist bench for 15 seconds every 5 minutes. So results of the study indicated that the two accessions are easy to root. There's actually no need for rooting hormone. There was a significant difference, though, in length of longest roots between selections. So the compact form actually formed longer roots than the prostrate form. And with this result, we're looking at doing evaluations for this particular species as a potted plant and as a landscape plant. Now we go on to the next species. We're looking at Pacific lovegrass or Aerogrostis deflexa. This dryland species is endemic in the dry leeward forest of Lanai and Hawaii Islands. The selection that we got were actually collected from the island of Hawaii through USDA NRCS Plant Material Center. So we're looking at this particular species as a potential accent plant in landscapes as well as a container plant. Propagation of the species is mainly done by seeds, but there hasn't been any studies looking at propagation by division. So we looked at asexual propagation by division as one of our studies, as well as landscape. We're also conducting landscape trials. So for the propagation study, we tested whether bare root clumps would benefit from soaking in water or in hormone dips. So our treatments was overnight soak of the clumps, which is about 10 to 15 stems with new shoots, in either tap water, 1 to 20 dip and grow, and 1 to 10 dip and grow. We use this concentration of dip and grow because we were able to see in some species, in some Hawaiian grass species, it was beneficial for it. And in addition to the soaking treatments, we also had the no-silk treatments and we rooted these clumps in a mist bed for 45 days. What we found out after 45 days is actually soaking the clumps was detrimental to the plant. So all soak treatments experience 100% mortality. And the unsoaked clumps, which is just bare root clumps and then just placed directly in the mist bench, had about moderate success, about 70% rooting. And based from this study, we kind of concluded to still use, it's still best to use seeds for propagating this species. And then our third species for the landscape component is looking at Alima. So Alima is a very highly variable species that's found from the coast up to 1,980 meters above sea level. And this particular species is found on all main islands. And it has been used in landscaping as a ground cover or as a hedge. But it hasn't been used as a potted flowering plant or as a container plant yet. So we're looking at those evaluations. And propagation is usually by seeds or cuttings. But if you want to maintain, with the use of seeds, you actually have seedling variability. So if you want to maintain the same characteristic of a certain selection, you want to produce cuttings out of it. So the research on Alima, we're doing collection and characterization of selections, as well as propagation and landscape trials. So since Alima is a very highly variable species, it provides an opportunity to do selection work. There's currently only 2 main cultivars of horticultural cultivars of Alima. This is black coral and Kaneohe gold. And our germplasm collection, we've been collecting germplasm from the different islands. So we have 6 wild collected plants from Hawaii, and then 3 cultivated from nurseries, and then from gardens. We have 2 seedling selections so far from Molokai, 6 cultivated types from Maui, basically coming from nurseries and Maui Nui Botanical Gardens. On Hawaii Island, we just collected about 17 accessions of, basically it's 8 prostrate types and then 8 shrub type plants, and then 3 cultivated. On Oahu, we collected 3 cultivated plants, and then we're planning on doing some wild collections in early January. So the plan is to do some collection of the germplasm and then grow out and then select for accessions that have potential use as in landscaping, as well as for potted flowering plant. So all of these 3 selections are currently being, or species are currently being tested at the UH Manoa campus. So we started a native plant trial garden in front of Queen Ilokalani Student Services Center. So this is at Barney Circle. And we initiated construction of the irrigation and then the new planting beds in summer of 2017 in collaboration with the UH Manoa Buildings and Grounds. We got design inputs from the campus landscape architect, Janet Gilmore, and Andy Kaufman. Then we got funding from the Women's Campus Club grant of about $1,000 for irrigation. And for planting as well as for maintenance, we're looking at tapping into students taking directed research. So as I mentioned before, the 3 selections that we've been looking at for Barney Circle are as follows. So we have Pacific Lovegrass, which is a selection we got from NRCS, and Aveo, the compact selection of Aveo. And then we had actually did some initial crossing and then created a hybrid of Alima, and we're trying to test that if that can have some useful some use in landscaping. And this is the planting plan for the Barney Circle. So the inner circle is the existing Pohina-hina, and then the outer circle is where you have the new selections being grown for testing. So this is us during the planting in early August. And this is some more pictures of Barney Circle. We already planted the plants over there. And then this was taken just, I think, Monday or Tuesday this week. So as you can see, some of the plants are already growing, especially the Aveo. So a closer look shows you Aveo growing very vigorously, very nice, compact. The grass is growing along, and the Alima seems to be growing as well, but it's a little bit slower pace as Aveo. So next steps for this trial garden. So we're going to install a Wi-Fi enabled soil moisture sensor to monitor soil moisture, because we want to know how much water do these plants really need. We're also looking at collaborating with Dr. Cheng to monitor the pest issues that we'll encounter while growing these plants. And hopefully after 6 months, we'll have evaluations. And if you guys are interested in trialing some of these selections, we're also open to looking at collaborators and sites, because the more sites that we have for evaluation, the better, because the better data that we have in terms of performance of these selections. So that's the landscape component. Now I go on to the indoor component. There's not much studies that have been done in terms of indoor potential of native Hawaiian plants. And our program focuses specifically on peperomia and Carax species. Right now, we are doing indoor trials and propagation studies specifically looking at peperomia, the Hawaiian peperomia. So Ala-ala-vai nui, or peperomia, the species of peperomia, we have about 25 species here in Hawaii, 2 that are indigenous or found here and found elsewhere. And then we have about 23 endemic. So endemic peperomia meaning it's only found in the Hawaiian Islands. And the diversity of species that we have in varying shapes and colors provide the potential for selecting plants for indoor or office houseplant, or as a green wall, indoor green wall. And peperomia in general are succulents, and so they kind of require not much water in watering. So actually, the plants that I am testing right now, we're just watering it under air-conditioned rooms. We're only watering it like once a week. So we're building a germplasm collection of peperomia. So far, we have collected about 37 plus accessions, representing about 10 species out of the 23. So about half of what's endemic and native. And we've been collecting from both cultivated and wild sources. So people who have been collecting these peperomias as a hobby, we've been collecting those materials. And also we've done some wild collecting just this summer in Kauai. And actually this picture on the left shows you the diversity of the peperomias that we've collected from Alakai Pihea Trail. So the 4 species that we're currently trialing for indoor use are these species. So we have Peperomia cookiana on your left, and then upper left, and then upper right is Peperomia leptostachya, which is the ones that are currently available in the market. Peperomia samvichensi is one promising species because it has a really nice color on underside, like a reddish tone. And then Peperomia oahuensi is another species that we're evaluating. So just as a, I guess, an update of what's happening right now with the indoor light treatment. So we're kind of close to about 3 months now indoors. We're testing it at 3 different light levels. The first one on your left is the 4.2 moles per day, which is about low light for houseplants. So this is the lowest, usually the recommended, within the range of the recommended light levels for growing houseplants. And then we have 0.43 moles per day, which is your typical office conditions. And we have about 0.22, which is about half of the office conditions, which is our kill treatment, and basically looking at what species will survive in this like really, really low condition. And based on the pictures and based from our observation, it appears that P. sanvichensis is basically the most tolerant of all the 4 species that we're currently testing right now in terms of light levels. So I think it has some potential use as a houseplant. So in parallel with the indoor studies, we're also looking at propagating these species and how to propagate. So we have explored whole leaf cuttings and application of IBA to propagate Cookiana, Leptostachya, oahuensi, and samvichensis. Where we tested untreated versus treated with IBA. And the results indicate that leaf cuttings can be a viable means of propagating the species. So we have about up to 84% rooting for Cookiana, Leptostachya, and oahuensi. However, we got about 15% rooting in samvichensi, and we're speculating that the low rooting was actually due to 2 wet conditions. So we're going to test, we're going to do another study or trial or propagation study for samvichensis using drier conditions, maybe looking at humidity domes instead of a mist system, because these were actually rooted under a mist. So future steps for our project. So for the landscape, we're going to collect more plants to evaluate, characterize the accessions, and then add more trial sites for these selections, and then evaluate. And then once we evaluate these plants, we'll start propagating these promising selections for distribution. For the indoor project, we're propagating the collected species for the next round of indoor trials. We're going to refine propagation protocols, possibly explore tissue culture as a means to propagate the Peperomia species. And then once we've found the propagation protocols, then now we can distribute these plants to the industry. So I'd like to acknowledge a lot of people, my grad students, Magoon Research Facility, the different arboretums and I guess agencies that have helped me in terms of getting plants and getting permits for collections. And this project was funded by the Hatch Project and the New Germplasm Grant, as well as the UH Women's Campus Club Grant. So that's the end of my presentation. If you have questions, let me know. Thank you.
Orville, can you hear me? This is Cynthia.
Yes.
Okay. I had a question about the Aveo. Is it really prostrate to the ground? Is it acting like a ground cover?
The prostrate one is really like hugging the ground, but it's not enough to like cover the whole the whole ground. It may be useful for like if you have like a rocky landscape,
Okay.
but not really for like really dense cover. If you're looking at really dense covering, then I would suggest the compact form, which is a little bit more. It's less than a foot high, but it's slow growing as well.
Okay, thank you.